Vascular system. In the early embryo, one can identify the process of early blood vessel formation in the wall of the umbilical vesicle. Extraembryonic mesodermal cells differentiate into angioblasts and begin to cluster. Cells innermost to the clusters undergo apoptosis, leading to the formation of blood islands. Some angioblasts flatten to form endothelial cells which line the early blood vessels. Other endothelial cells differentiate into hemangioblasts and form blood cells. Individual blood islands then coalesce through the process of vasculogenesis to form a network of primordial blood vessels from which additional vessels sprout by the process of angiogenesis. This rapid growth of vessels provides the necessary means by which nutrition and oxygen can reach the developing embryo. 3 paired veins drain into the early embryonic heart. The cardinal veins return poorly oxygenated blood from the cranial and caudal portions of the body of the embryo. The vitelline veins return blood from the umbilical vesicle. The umbilical veins carry well-oxygenated blood from the placenta. As the heart continues to develop, the pairs of vessels undergo remodeling to develop the definitive venous connectivity to the heart. For example, the right umbilical vein and the cranial portion of the left umbilical vein disintegrate. The left vitelline vein also regresses, while the inferior vena cava forms from derivatives of the cardinal veins. The brachiocephalic veins and the superior vena cava develop from the anterior cardinal veins. Let us now consider the vitelline and umbilical veins. The vitelline veins, carrying deoxygenated blood from the umbilical vesicle, and the umbilical veins, carrying oxygenated blood from the placenta, enter the sinus venosus, the venous end of the developing heart. The developing liver can also be seen. In this image, the vitelline veins have begun to form the hepatic portal system, while the umbilical veins can be seen on each side of the developing liver. As development continues, the umbilical veins lose their connection to the heart and empty directly into the liver, and the right umbilical vein regresses. The left umbilical vein becomes the true umbilical vein. The ductus venosus, a large venous shunt, also develops in the liver, connecting the umbilical vein with the inferior vena cava. Remnants of the right vitelline vein include the hepatic portal vein and the splenic and superior mesenteric veins. The anterior and posterior cardinal veins, the earliest veins to develop, comprise the main venous drainage system of the embryo. The common cardinal vein provides a conduit for the vitelline and umbilical veins to the sinus venosus. To supplement the posterior cardinal vein drainage, the subcardinal and then the supracardinal veins develop. The subcardinal veins interconnect to each other through the subcardinal anastomoses and connect with the cardinal veins through the mesonephric sinusoids. The supracardinal veins develop between the subcardinal and cardinal veins and interconnect with the former through anastomoses, shown in green. The inferior vena cava develops from 4 main segments of these veins. The hepatic segment is derived from the hepatic vein and hepatic sinusoids, a prerenal segment from the right subcardinal vein, a renal segment from the subcardinal supracardinal anastomosis, and a postrenal segment from the right supracardinal vein. The complexity of transformations that occur may result in a number of variations in venous patterns that are found in the adult. The pharyngeal arches form during the 4th and 5th weeks and develop a blood supply, the pharyngeal arch arteries, which originate from the aortic sac and terminate in the dorsal aorta. Although 6 pairs of arteries form over time, the first 2 pairs contribute to only minor structures, including the maxillary arteries and the stapedial arteries, respectively. Typically, the 5th pair of arteries is only very rudimentary or does not develop at all. The arteries of the 3rd, 4th, and 6th arches alter their position and contribute to several important structures. The 3rd arch arteries form the common carotid arteries and contribute to the internal carotid arteries. The left 4th arch artery forms a portion of the aortic arch while the right contributes to the right subclavian artery. The proximal portion of the left 6th arch contributes to the left pulmonary artery, while its distal portion forms the ductus arteriosus, a prenatal shunt. The right 6th arch artery proximal portion contributes to the right pulmonary artery, while the distal portion degenerates. A summary of the developmental changes is shown in this table. The location and transformation of the 6th arch arteries explain why the course of the recurrent laryngeal artery is different on the right and left sides of the neck. The nerves supply the 6th arch and are branches of the vagus nerve. The recurrent laryngeal nerves also hook around the arch arteries to connect with the developing thyroid gland. On the left-hand side, the recurrent nerve loops around the forming ductus arteriosus as it develops from the distal left 6th arch artery. Although the ductus arteriosus involutes after birth, a remnant of it, known as the ligamentum arteriosum, persists, with the nerve maintaining its position. On the right-hand side, the distal portion of the 6th arch artery disintegrates, leaving the nerve to hook around the right subclavian artery, a derivative of the 4th arch. The fetal circulatory system serves the needs of the fetus in utero, but must adapt to the requirements of the neonate. Prenatally, the foramen ovale acts as a shunt between the right and left atria, allowing a majority of blood to bypass the developing lungs. The ductus arteriosus similarly shunts blood from the pulmonary trunk into the descending aorta. The ductus venosus, the 3rd vascular shunt in the fetus, allows about 1/2 of the oxygen-rich blood from the umbilical vein to bypass the sinusoids of the liver and pass directly into the inferior vena cava. This animation demonstrates the flow of high, medium, and low oxygen blood through the body of the embryo, including the fetal shunts. At the time of birth, the blood supply from the placenta ceases and the infant's lungs expand and function. This lung aeration results in a significant decrease in the pulmonary vascular resistance and a concomitant increase in pulmonary blood flow. These changes lead to the pressure in the left atrium increasing in comparison to the right atrium, resulting in closure of the foramen ovale. The ductus venosus closes so that all the blood entering the liver passes through the sinusoids. The increased oxygen level of the blood entering the aorta triggers a contractile reaction in the ductus arteriosus, resulting in its closure, most typically over a period of between 24 and 96 hours.